Hello, and welcome to Skidmore's Northwoods, a 150-acre temperate deciduous forest located north of the city of Saratoga Springs. In this 300-acre forest, a complex series of trophic interactions is constantly shifting and evolving. Let's take a closer look. As winter approaches, the leaves of most deciduous tree species wither and fall to the ground, creating a banquet of brown organic material that forms the bottom of a complex food web based on its breakdown. The foundation for this web is made of decomposing bacteria and fungi, who are responsible for the chemical breakdown of plant materials. Microarthropods, such as Collembola and Eurybatids, are detritivores that provide a critical link from the brown to the green food web. These organisms feed on microscopic bacteria and fungi, thus linking the detritivores into higher food webs where they are consumed by macroarthropods such as mites, pseudoscorpions, and spiders. At the top of the brown food web lies this magnificent amphibian, Notothalmus viridensis, the F stage of the red-spotted newt and a voracious predator of microarthropods. In this study, we sought to link the green and brown food webs by comparing F density with rates of organic decomposition. We hypothesized that this food web will be regulated in one of two ways. In the case of bottom-up regulation, we expect to see the abundance of microorganisms having a positive effect on higher trophic levels. On the other hand, in the case of top-down regulation, we expect to see predation from Fs having a negative effect on invertebrate abundance, but a positive effect on decomposer abundance, as evidenced by higher rates of leaf breakdown.
We determined F densities by performing transects on the trails in the Northwoods. Fs within 6 feet of each edge of the trail were counted, and every 10 meters the F counts were recorded for the transect. To measure soil respiration, around 8 grams of soda lime was measured into small plastic containers. These containers were then brought into the Northwoods and placed in random locations throughout the woods. To place these containers, a 30 cm by 30 cm area underneath the container had to be cleared of freshly dropped leaves, which were collected and placed in a plastic bag. A respiration chamber was then placed over the container and pushed into the ground to create a seal. This was done to ensure that only the carbon dioxide being respired by the soil was being measured. These containers were left out for 48 hours, collected and masked. The gain in mass of the soda lime was used as a representation of how much carbon dioxide was respired. We searched through the collective leaves for macroarthropods, which were then masked. The Northwoods had previously been surveyed by Ness and Warren, who divided the forest into 35 grid cells, each 1/10 of a minute latitude by 1/10 of a minute longitude. Arthropod mass per grid cell, F density per grid cell, and average soil respiration per grid cell were compared using linear regressions run on Microsoft Excel.
Here we have F abundances recorded by handheld GPS units plotted on a map of Northwoods. The red dots represent where Fs were observed. Larger red dots mean greater amounts of Fs. You may also notice the clouded regions of the map. These represent F hotspots. To compute F density per grid cell, this map was superimposed on the gridded map from Ness and Moore in 2008, using a projector and a smartboard. Distance of path per grid cell was then calculated with the use of Google Earth's ruler tool. This is a map of the average F abundance per meter of path per grid cell. As you can see again, the red regions represent grid cells in which the F abundance was as high as 1/10 to 1/4 of an F per meter of path. Blue cells were cells in which either no Fs were observed or the F density was lower than 1/100th of an F per meter of path in that grid cell. Here we see that no significant relationship exists between F density and invertebrate abundance. The p-value here was in excess of 0.41. However, since some of the data collected on invertebrate density was less than 0.004 grams and could easily be due to air, we can remove these data points to find an interesting power curve that has a high R-squared value. In this case, we see that invertebrate abundance is high when F density is lower than 3/100ths of an F per meter of path. When F abundance is higher than that, invertebrate abundance drops dramatically. There was no significant linear relationship between F density and soil respiration. The p-value here is in excess of 0.6. There also wasn't a significant linear relationship between soil respiration and invertebrate abundance. The p-value here is greater than 0.36.
Our results don't show any patterns to indicate that either top-down or bottom-up regulation is the driving force behind this food web. Only that F predation can significantly influence arthropod communities. However, in previous work, it has been shown that both bottom-up and top-down control can simultaneously act on a system, and we believe that to be the case here in Northwoods. In future work, it would be interesting to see how the removal of one part of this food web, such as the Fs or the arthropods, would influence the community as a whole. So, until next time, thanks for tuning in, America. Alright, fine, we're done. Now help me bury this body.